So Gregor Mendel is one of my more favorite scientists, and he was an Austrian monk in the 1800s, and he did his work around, I think maybe around 1850, 1860, and so he recognized patterns and analyzed physical traits and how they were inherited from parents to offspring, and he did this using pea plants. So when he began his work in the mid-1800s, he acknowledged that both sexes, the male and the female, contributed equally to a new individual. He tested various combinations of traits, and these combinations are what led to reproductive success that were passed on to further generations. And if you see here, some of the pea plant traits that he used, he noticed some were recessive and some were dominant. The recessive traits were like short stem length, a wrinkled seed pod, and like the color green was recessive. And so he crossed these with the dominant to see, where are we going to pass these on? When we cross it with the dominant, does the recessive just completely go away, or does it crop back up in future generations? We'll start with what's called a one trait inheritance. And so this is coming from your original parents, and it's noted as the P generation, and P standing for like your parental generation. So then you'll have your first generation of offspring when those are combined. So that's your F1. So and then you'll have an F2, which is your second generation. So he started with crossing green pod plants with yellow pod plants. So he crossed a green and a yellow pod. And so this was your P generation right here that were crossed. Then what he ended up with, what was his F1, his first generation, he noticed he got a green pea pod. So then he was like, well, what happened to the yellow ones? Did they disappear? Mendel used a Punnett square, and that shows all the possible combinations of egg and sperm that an offspring may inherit. So when the first generation allowed to self-pollinate, then your second generation, your F2 generation, were 3/4 green and 1/4 yellow. The F1 generation here had passed on that yellow pod when it was crossed. So Mendel reasoned that there was a 3-to-1 ratio that was possible only if your F1, your first generation parents, contained two separate copies of heritable factors. One had to be dominant and one had to be recessive. So factors that separated when gametes were formed, and then each gamete carried only one copy of each factor. So Mendel used a one-trait test cross to see if the first generation carriers had a recessive factor. So he took his F1 generation green pod plant and he bred it with a true breeding yellow pod plant. So he reasoned that half the offspring would be green and half would be yellow. So he had previously hypothesized that those factors will segregate when gametes are formed, and so his hypothesis was supported by this. So meiosis explains Mendel's law of segregation and why only one gene for each trait is in a gamete. Remember, a gamete is an egg and a sperm. So when fertilization occurs, the resulting offspring again then have two genes for each trait, one from each parent. So here is a picture of homologous chromosomes. So alleles are going to be an alternate form of a gene. So if you notice right here in the picture, we have this green G, and it's dominant, so it's a capital G. Then we have a yellow one here, and it is a lowercase g, so that is going to represent a yellow color. So a dominant allele will mask the expression of that recessive. So if these two pair up, we're going to have this gene here, but it's not going to show up. The dominant one is going to mask it and cover that recessive one up. So for the most part, an individual's traits are going to be determined by the alleles that are inherited. So all of these alleles, right here, right here, right here, these alternate forms of genes, they occur on a chromosome at a particular location called the gene locus. And so if it's plural, it's going to be loci. These terms are going to be really important for you to even understand any of the concept in this chapter. So we use this word genotype versus phenotype. Genotype is going to be the exact alleles. It's the specific alleles the individual has and received at fertilization. So it's going to be represented with the 2 letters or 4 letters. So it can be expressed like if you want to write the genotype down, you would say this one is 2 capital T's or it is a capital T lowercase t. That's the specific genotype. Now in the genotype, if it is homozygous, the word homo means the same. And so that means it's 2 identical alleles. So right here, that would be homozygous. And 2 lowercase, those would be the same. So they're 2 identical. This, a capital T and a lowercase t, that is going to be heterozygous. That's 2 different alleles. Now, if we look, let's go back to homozygous. If we look at homozygous dominant, it's going to mask the alternate allele. So it's going to mask the recessive. If it is homozygous recessive, then that expression will actually show up. So you won't see the dominant allele. So if we're looking at capital T representing that it's a tall pea plant, and we know that's dominant, the only way that we would see a short pea plant is if we saw the homozygous recessive 2 lowercase t. Then we would know that would be a short pea plant. When we say the word phenotype, I want you to notice it starts with pH. That sounds like physical. Physical starts with pH. So the phenotype is the physical appearance of the individual. It's what you exactly see. Like your phenotype would be your hair color or we really need to understand these terms. So if we look at the genotype, it's going to be represented by the allele combination. So here your genotype would be 2 capital A's, which would be homozygous dominant. Homo meaning 1, and so, or the same. And so we have both of them are the same. They're going to be dominant because they're capital letters. This is saying that it is normal pigmentation. You're going to have color in the skin. If it is heterozygous, hetero means different. And so you have 2 different allele combinations here, a capital, a dominant, and a recessive. You're still going to have color and pigmentation in your skin because that dominant one covers up that recessive. It masks it. Then if you have homozygous recessive, recessive is the lowercase, and it's homozygous, homo meaning the same, so 2 recessive. That's when you're going to see albinism, when there's not going to be any color or pigmentation. Okay, so let's look at this chromosome right here. A chromosome will have many, sometimes even thousands of genes, and here's where they're represented here, these alleles. This is going to be an example having 4 pair. And so if we look at the first, we have a heterozygous, hetero meaning different, we have a dominant and a recessive. Then the next pair, we have capital R, lowercase r. Again, that's heterozygous trait. Again, we have another heterozygous, a capital S, lowercase. And this one, we have another heterozygous, even though that we have the recessive being shown first. So with this individual is heterozygous for all traits on this chromosome. Let's look at this one trait test cross example. So we want to know that which phenotype will be absent in the F1 generation. Here's some things that
so our question is, we in a one trait test cross example, we have a homozygous dominant individual. So let's just use capital B. So homozygous, they would have to be the same because homo means the same, dominant. Then we're crossing with a hetero or homozygous recessive. So we'll use lowercase b's for recessive. That's homo, that would be the same. So which phenotype will be absent in the F1 generation? So that's your first generation. So we can put these on the side or these on the top. It doesn't matter. So let's start over here. We'll put these on the side. Your capital B's, your dominant, then let's put our recessive on top. It could be either way. So to set this up, you're going to come across. You're going to bring this one down. And it always looks better if you write it with the capital letter first. Okay, so let's go across. Let's bring this one down. Same thing, let's go across. Bring this allele down. Cross, bring this allele down. Now, notice we have 1, 2, 3, 4 heterozygous. So we are coming up with 4 heterozygous right here. So this wants to know which phenotype will be absent in this. So for it to be absent, it must be recessive. So we're not seeing this phenotype. Let's just use, say we're talking about mice, that we're using black mice. The capital B stands for black. So this would be, say, a white mouse. And so we don't have any of the recessive phenotypes showing up. So all of these would have to be black and none of them would be white. So our recessive phenotype, which would be 2 lowercase b's, is absent, but only in this F1 generation.
Now, two-trait inheritance is a little bit different. So Mendel crossed tall pea plants with green pods. So our two different traits, we're talking about the height and also the color of the pod. So now we're going to have 4 alleles, and then he crossed it with a short plant with yellow pods. So both of those are going to be homozygous recessive. And so now you're having 4 alleles in there. So his F1 plants, his first generation plants, show both dominant characteristics, tall and green pods. And then there were 2 possible results for the F2 generation. So that was the next generation of cross. Now, when you're setting up a Punnett square for a 2-trait cross, notice this Punnett square looks a lot different. And so instead of having 4 squares, you're now going to have 16 squares. And it's a little bit more complicated, but it's not. You still take each allele, and you, like on the, say if we're saying the female, we're going to put each allele at the top. We're going to put each allele at the male going down the side. And then you're going to take these same 2 across, this T and the G. You're going to come across with it. You bring your T over, your G over. Then you bring a T and a G down. Now, all they did is they put their T's together and their G's together, because then when you go down here to read your phenotype, it's much easier to read the phenotype. So when Mendel was looking at all of his results, he came up with his second law of independent assortment. So this is where alleles of different genes will segregate or sort independently of one another. And this will give you all possible combinations of factors that can occur in different gametes. Mendel's laws apply to a lot of like genetic counseling and things like right now, because genetic counselors may already know that there's a pattern of inheritance, and then they can predict the chance that a child born to a certain couple would either have an abnormal or a normal phenotype. So with autosomal recessive disorders, a child can be affected when neither parent is affected. A heterozygous parent are going to be the carriers. So parents can also be tested before having children. This one's a little bit different because it's not just the father that's a carrier, but the mother actually has the disorder. But it does show you this different pedigree to how some of the children can be affected and some may not be. Here we're needing to figure out what the genotype is of the father.
Okay, so it says that albinism in snakes is a recessive trait, and an albino does not have color. So we're going to set up our genotypes as a capital C. This is going to be a homozygous dominant. So this is a genotype. This is definitely going to have color. Heterozygous would be a capital C, lowercase c. That is still going to have color. To be albino, you are going to have 2 of your recessive alleles to show up. So you would have to have 2 lowercase c's to be albino with no color, basically no color. Okay, So to set this up, it tells us that the female albino, we know she's an albino, so we're going to set this up with 2 little C's. Now, you're going to have to kind of play around to figure out your Punnett square of how this would work. So let's start. We need to find the father, and we know that some of the offspring were colored and some were albino. Okay, so if we say that his genotype is going to be homozygous dominant, if we put that in here and we work this Punnett square out, bring that down, bring this across, bring that across, that down, this across, that down, you're going to get 4 heterozygous. Heterozygous has color, so it cannot be that one. So we're going to mark those out. Now, if we did say that he was completely albino and we put these little C's in here of no color, and we bring those across, what are we going to get? We're going to get 4 albino. And that's not going to work because some of the offspring did have color. So it can't be that he's an albino. He has to be heterozygous right here. So that would be a capital C, lowercase c. So if you bring it down, capital C, lowercase c, a little c, little c. So right here you already have one with color and one albino. So the father's genotype, he has to be heterozygous.
A child can be unaffected with the autosomal dominant disorder, but both parents would have to be heterozygous. And so if you notice at the top up here, we have the mother has the disorder because it is dominant disorder, and so does the father, but they are both heterozygous. And then if you follow the pedigree down and over to the right, you notice that those 2 daughters inherited both recessive traits. And so when they married someone with a recessive trait that was homozygous recessive, then therefore none of their children that do not have the condition, nor is that dominant gene passed on. We've only previously been talking about complete dominance, and where you either have that trait or you don't have that trait. This is incomplete dominance, where a heterozygote has an intermediate phenotype. So phenotype being that physical trait. So the easiest way to explain it is if you cross a red flower and a white flower, incomplete dominance, you would end up, if it was heterozygous, then you would end up with a pink flower. So that would be your intermediate phenotype. And then if you take the next generation and cross it again, that's when you'll start showing up with several, like you'll have a 50% chance of it having that intermediate phenotype, and then you'll have one dominant red and then one dominant white. Our ABO blood typing has 3 different alleles. If you have type A blood, you have an A antigen on your red blood cell. Note how it's represented here with a capital I and a capital A. If you have a B blood type, that means you have a B antigen on your red blood cell. If you are neither, that means you're a type O, and so it would be represented here with 2 lowercase i's, would be type O. Now, A and B can be co-dominant, so they are dominant over type O blood, but they are co-dominant, and they would be represented as a capital A and a capital B. And people can have like a type AB blood.
Okay, Edward has Punnett square completely backwards, and we know that type O blood does not have the A or the B antigen, and so it's represented like this with a superscript of a lowercase I and a lowercase I. So this would give you type O blood. So I would work it backwards. I would put the child in this bottom square here with an O type blood. So it would look like this. Now it gave us the information that the man is blood type A. Well, let's move these around first, and then we'll get to that. So let's do this. We'll take the male, let's bring it up here. And then we're going to take the female genotype and let's move it across to where it started from. So it would look like this. Okay, so now if we're going to say this is the male, we would say if he has a blood type A, he would have to have superscript of a capital A. So this means he's heterozygous, that he's not homozygous. If he was homozygous, this would be 2 A's, but this would still represent blood type A. And it says the woman has blood type B. Well, is this also a representative of blood type B? Yes, because she would be heterozygous. So if we wouldn't even have to fill this in. So you would have a B and an A, and so we know that would be blood type AB. We would know that, I'm kind of writing this backwards. I should write it the other way. This is another blood type B. Then if we come across and we come down, and technically you should always write this on this side of that one. So this would be blood type A, and then if we came across, this would be blood type O. So is it possible that a man with A and a woman with blood type B can have a type O child? Yes, it is possible.
Word poly. Poly means many, and so this is polygenic inheritance. This is when a trait is going to be governed by 2 or more sets of alleles, and each dominant allele is going to have a quantitative effect on a phenotype, and so the physical appearance of it. And so you're going to, if you graph this, you're going to get a bell-shaped curve. And so there's also things that environmental effects that go into a lot of these polygenic inheritance traits. So polygenic traits do not follow the patterns of Mendelian inheritance. Many polygenic traits are going to be influenced by the environment and are also multi-factor. So height is a very good example. It's controlled by at least 3 genes with 6 different alleles. So if you're dominant for all of the alleles for height, then you would be very tall. Skin color is an example of a polygenic inheritance trait, and the environment definitely influences this because our skin responds to like UV radiation, the melanin that's produced in there that gives it the darker color, and human production of melanin in skin increases closer to the equator to protect the skin from radiation. And if you look at this chart here at the bottom of it where it says the number of dominant alleles, and so you can see that there's many different combinations here, and again skin color is an example of this. Eye color is another example here, and multiple pigments are going to be involved in determining eye color. Just because you have blue eyes, you may have a lot different pigments in there than someone else with blue eyes. And so there's many, many different shades of blue eyes and brown eyes and green. And sometimes people with brown eyes have a little bit of green pigment in there too. Sex-linked inheritance means that it is going to be a disease or a disorder or some type of allelic trait that is going to be carried either on the X or the Y chromosome. Females have 2 X chromosomes and a male has an X and a Y chromosome. Colorblind is going to be a recessive X-linked inheritance disorder, and so that means it has to be carried on the X chromosome. If you look at the key over here, normal color vision, you're going to have a capital B. To be colorblind, you're going to have that lowercase recessive. This female on the top right is going to be a carrier, but will have normal vision. The father on the top left over there also has normal vision. For a male to be colorblind, you have to have the lowercase, the recessive B on all the X chromosomes. They only have 1 X chromosome. So if you look in the far bottom right of the Punnett square, this is going to denote a male with color blindness. So if it is an X-linked recessive disorder, this is going to mean that the disorder will show up on the X chromosome, not the Y. And so for it to be recessive, if it's a female, females have 2 X chromosomes. So it would have to show up on both of their X chromosomes. But we know that male only they have an X and they have a Y. So it would have to, if it shows up on their X chromosome, then that's the only X that they have, so the recessive disorder would show up. And so this means that a male is always going to be homozygous for an X-linked trait. And females who have the condition inherited the mutant allele from both their female parents and their male parents. And conditions appear to pass from male grandparents to male grandsons with X-linked recessive disorders.
Let's look at this X-linked disorder. X-linked has to be on the X chromosome, not the Y. It says a mother with Alport disease whose father was unaffected marries a man that is also unaffected. How many sons in each pregnancy can be born with Alport syndrome? We got to look at the key factors to look at. First of all, it says, are we going to need to know, is the mother heterozygous or homozygous? Well, her father didn't have it, and so she would have to be heterozygous. So to be heterozygous, she would have, her genotype would look like this: an X with a capital A and an X with a lowercase a. Now, the father of the son is also unaffected. So his is going to be XY, and so we know he would have to be homozygous recessive is what he would have to be. So his X chromosome would have to be a lowercase a, and there's nothing carried on this Y chromosome. So let's set this up in a Punnett square. We'll put the father on the top part of it. So we say that it is going to be represented. He does not have it, so that's recessive, lowercase. Nothing goes on the Y chromosome. The mother, we said, was heterozygous. So, and because it's a dominant disorder, if you're even just a carrier of it, then you're going to have that disease. So let's go this one across. We're going to bring this one down. We're going to come across, capital X, capital A. We're going to bring this one down. There's your Y. Then we're going to come over here. We're going to go across, lowercase, because we're bringing it down. Now we're bringing this one across, and we're going to bring this one down. Now we need to know how many sons in each pregnancy. Well, this is a daughter, so we don't look at it. This is a daughter, we don't look at it. This daughter would not have it. This daughter would. Now here's a son. This son, yes, has it. This son, no, does not have it. So how many sons would we have with Alport syndrome, or a possibility? We could say one son would be the possibility.
Look at an X-linked dominant disorder. The daughters of the affected males will have the condition, because if the male has it on his X chromosome, and we know it's a dominant trait, it's only going to have to show up on that daughter, only on one of her X chromosomes, for her to have the disorder. So affected females can pass condition to female offspring and male offspring, but the daughters of affected males will always have the condition. So if it's the Y chromosome, only a few disorders. They're present only in males and are passed to all male offspring, but not any female offspring, because of course females don't have a Y chromosome. Gene mutations are going to be basically a change in the sequence of bases in a gene. When there is a mutation, then most of them, there's no observable effects, because many mutations go undetected. There's different types of mutations and how they occur. They can be point mutations or frame shifts, which we'll get into that a little bit later in the next chapter. So chromosomal mutations are going to be changes in the number of the chromosomes, and we know conditions like this are going to be like Down syndrome, Turner syndrome, and Klinefelter syndrome. So in general, mutations are going to be relatively uncommon, because there are DNA repair enzymes that often fix these errors. So with genetic counseling and things, we can test for proteins. And some disorders that are going to be caused by a missing enzyme, we can test for the quantity of the enzyme that's produced. So when we test DNA, we are looking at genetic markers. And a genetic marker is going to be a known abnormality in a gene sequence that can be linked back to a genetic disease. When we're looking at those genetic markers, we're going to take a person's entire genetic profile, and that profile is going to be a complete genotype, including all their mutations.